everyone, welcome to the course on medical biomaterials. We will continue on the topic of biofilms. Biofilms, as I said, is the most serious issue in the area of biomaterial implants devices. Most of the biomaterials or implants fail because of formation of biofilms and which reduces the life of implants or even devices. Now, what is this biofilm? It is a population of microorganisms concentrated at a surface, okay, and it is surrounded by extracellular polymeric substance. So, there is a matrix that is formed which sort of protects these bacteria and thereby they survive for a longer time. They are able to prevent getting killed by the antibiotics. So, some of them even develop antibiotic resistance and so on. So, this biofilm may contain live or dead cells, it will contain proteins, it will contain sugars, polysaccharides, metabolites, even quorum sensing signaling molecules. Okay. So, most of the gram positive, gram negative bacteria form biofilms in various parts of implants. It could be a dental implant, it could be a cardiovascular stent, it could be a urethral stent, it could be a orthopedic material and so on. So, E. coli, Pseudomonas, Staphylococcus aureus, Staphylococcus epidermis, Proteus mirabilis, Klebsiella, Enterococcus, Streptococcus, so many different types of organisms that can form a biofilm. Okay. There is something called persisters. These are cells, bacteria which are in biofilm and they start remaining for a longer time, they become persistent. Okay. That is a small subpopulation of bacterial cells that will be dormant. So, they become extremely tolerant to antibiotics. So, they acquire this antibiotic resistance, hence they persist in the biofilm. Okay. So, these are a phenotype expressed by almost all the bacteria including major pathogens. This can lead to chronic and relapsing infection. Okay. So, because the biomaterial has been placed, some bacteria survive for a longer time and they become immune to antibiotics also, that is persister cells. They can tolerate high doses of bactericidal antibiotics. Okay. A small fraction of bacterial population responds and others form resistance or tolerance towards these antimicrobial agents. Okay. So, why do they become? They change their phenotype. There is an exopolysaccharide matrix that is formed on the surface. It prevents the diffusion of the antibiotics right to the bottom of the biofilm. So, many reasons happen. Okay, we will talk about some of those reasons as we go down the class. So, why do they get antibiotic resistance? Okay, this is called biofilm associated antibiotic resistance. There are many factors which lead them to have bacterial resistance. For example, the presence of exopolysaccharides and glycocalyx. Okay. This glycocalyx is nothing but is a glycoprotein polysaccharide covering that surrounds the cell membranes of some bacteria, okay, and even cells. This is nothing but strands of sugars and proteins bound together. So, it prevents the diffusion of the antibiotics. Okay. There are enzymes which are produced by the bacteria and they are called antibiotic degrading enzymes. So, these enzymes, as soon as the antibiotic is released into the system, they come and degrade the antibiotics. So, the antibiotics are no more effective. There are extracellular DNA which also affects which leads to antibiotic resistance. There are something called efflux pumps. Many bacteria possess this particular protein called efflux pump, and these pumps out whatever foreign material that enters the bacteria. It could be an antimicrobial agent, it could be an antibiotics, it could be a dye, it could be a toxic chemical. So, these efflux pumps throw them out, thereby it prevents the killing of the bacteria. Quorum sensing. Many bacterial species release a chemical called a quorum sensing molecule. It is a metabolite just to inform each other that there is a certain cell population density. Okay. Quorum sensing helps in the biofilm formation. Basically, quorum sensing molecules help the bacterial population that means it has reached a threshold population that they can form a colony. Okay. So, all these factors have an influence on the bacterial species gaining antibiotic resistance, and we will look at each one of them little bit, and this leads to biofilm associated infection also in biomaterial. So, the factors that affect bacterial resistance, we can group it into biochemical factors, we can call it molecular mechanism, we can call it altered host factors. Biochemical factors, like I mentioned, exopolysaccharides that are produced, antibiotic degrading enzymes that are produced, extracellular DNA, efflux pumps, quorum sensing. If you look at molecular mechanism, there could be a gene transfer, lateral gene transfer or horizontal gene transfer. Sometimes the bacteria as soon as they settle on the surface mutates. Okay. All these can lead into resistance, antibiotic resistance. The third could be altered host factors, because the antibiotics could be under sub-MIC concentration. Like I said, the antibiotic has to diffuse through the biofilm matrix. So, the concentration of antibiotics in the interiors of the biofilm could be sub-MIC. Oxidative stress, the oxygen diffusion also is hindered in the biofilm because of the presence of the EPS. So, many of the bacteria can be undergoing oxidative stress. SOS response, chemical signals, toxin antitoxin modules that are produced, nutrients, amount of nutrients available inside the biofilm could be much less. So, they are under starvation condition. Temperature may be affecting, pH may be affecting, even the cell density, and finally the osmolarity. So, all these are called the host factors which may influence the bacteria to gain resistance, okay, in the biofilm. So, one needs to understand all these factors, and hence design surfaces which can prevent biofilm formation as well as which will eradicate persistent cells. So, let us look at some of these factors. The drug efflux pumps, like I said, these are large proteins which throws out whatever foreign material is inside. Okay. So, they pump out of the cell, mostly they are found in gram negative bacteria. They help the microorganism to regulate their internal environment by removing toxic substances. It could be antimicrobial agents, metabolites, whatever it is, these are called efflux pumps. There are many families of efflux pumps. You do not have to get very worried about these biological names, but you have to understand there are many families of efflux pumps. So, sometimes in the drug discovery, if they decide to design molecules for one family of efflux pump inhibitor, you could have several efflux pump families which you need to. One is called the ATP binding cassette superfamily, other is called major facilitator superfamily. There is a multi-drug and toxic compound extrusion family, small multi-drug resistance family, resistance nodulation division superfamily, drug metabolite transporter superfamily. So, even the metabolites that are formed when the drug is introduced, there are efflux pumps which can throw it out. So, a large number of families are there, and these are called efflux pump families which throw out any foreign metabolite that is present inside the bacteria. If you look at E. coli, E. coli has very well developed efflux pumps. This is a particular system, ACR-ABTOLC. This is a protein which consists of inner membrane transporter, a periplasmic membrane fusion protein, and an outer membrane channel. So, in the E. coli, these transporters right from the inside of the microorganism, they can throw the metabolite right outside into the extracellular. These are best characterized efflux pumps in E. coli and has been found to be overexpressed especially in clinical isolates, because clinical isolates develop a lot of drug resistance. The pump can export as you can see antibiotics like chlorometaphenicol, fluoroquinolines, fusidic acid, rifampicin, tetracycline, ethidium bromide, bile salts, SDS, surfactants like SDS, dyes, detergents, disinfectants, solvents, antibiotics. Large number of compounds can be thrown out by the this particular family. This family encodes genes found to be upregulated under growth in biofilms and exposure to several antibiotics. So, in the biofilm as well as when the E. coli is exposed to antibiotics, these genes which encode this particular efflux pumps get upregulated. So, they are very active and they are found in especially in clinical isolates. So, there are efflux pumps inhibitors. Okay. They are substances that inhibit the flux of substances mediated by efflux pump. So, what we do is, if you have a efflux pump inhibitor, it can go and bind to those proteins and make the efflux pumps inactive. Then the bacteria becomes okay, under any antibiotic. It can be easily treated under any antibiotic and they become very sensitive to those antibiotics. So, one strategy of dealing this type of resistance is you give the efflux pump inhibitor as well as you give the antibiotic as well. So, there are many inhibitors, thyroidazine and then PA beta N. These inhibitors could be used as enhancers of the antibiotics in the treatment of biofilm. Okay. So, for example, this particular compound contains this type of heterocycle with nitrogens and sulfur. This is a compound which has got arginine, naphthalamide here, you can see this. Okay. So, in addition to giving antibiotic, you can also give these one of these efflux pumps inhibitors, so that the biofilm can be eradicated. Okay. The next important activity in the bacterial biofilm is the quorum sensing. Now, this quorum sensing plays a very important role in the development of biofilm. So, what is this quorum sensing? When the bacterial population is very small and as the population keeps increasing, they start releasing a metabolite that is called a quorum sensing molecule. Okay. And when the concentration of this quorum sensing molecule increases, then the bacteria also realizes that their population has reached a threshold value and they start forming a biofilm on a surface. So, quorum sensing is almost like a signaling, and the signaling helps for these bacteria to change their phenotype from the sessile form to the fixed form. Okay. So, there is a lot of changes that happen as soon as they start settling down and form biofilm. So, there is a very strong connection between quorum sensing, signaling and the biofilm formation. Okay. This is almost like a social behavior of the bacteria. So, this quorum sensing is a cell to cell signaling, and this is controlled expression of specific genes in response to these extracellular chemical signals produced by bacteria themselves. Okay. And also the efflux systems have been implicated in quorum sensing regulation. So, the quorum sensing not only changes the phenotype of the bacteria from the sessile or motile form into fixed or biofilm forming form, but it also affects the efflux pumps. It also controls the expression of number of virulent factors as well as biofilm differentiation. Okay. So, there are three classes of quorum sensing systems have been reported actually. Okay. The first one is related to gram negative bacteria. There are two proteins, Lux and Lux I and Lux R. These are quorum sensing in gram negative bacteria, and the quorum sensing signaling molecule is called acyl homoserine lactones. This is the structure of these acyl homoserine lactones. Okay. So, there are a lot of these type of functional groups. Okay. This lactone ring is there and there could be longer chains. So, you can have hydrophobic molecules or very short hydrophilic. These are all called signaling molecules. These are produced by the gram negative bacteria. Okay. And at certain threshold concentration the phenotype of these bacteria changes because they realize their population density has reached a threshold value. If you look at gram positive bacteria, okay, they produced some oligopeptides. These are two component type quorum sensing in gram positive bacteria. These are small peptides equivalent to these AHL signaling molecule produced by gram negative. Gram positive produced small oligopeptides. Then we also have Lux S. These are encoded autoinducer 2 quorum sensing in both gram negative and gram positive bacteria. Okay. So, predominantly gram negative produces this acyl homoserine lactones, gram positives produce the oligopeptide. So, most of the percentage of bacteria is covered by these, and of course, we also have some Lux S which is produced, it is an auto inducer produced by both the gram negative and gram positive. Okay, let us look at how these Lux I and Lux R type of coherent sensing in gram negative happen. So, Lux I like protein is an auto inducer synthase that catalyzes the formation of a specific acyl homoserine lactone. Okay. So, the AHL is produced, okay, it goes to the extracellular space. So, at low concentrations the AHL gets dispersed, it diffuses freely through the cell membrane at high cell density. So, at high cell density they come back again and they bind to the Lux R. The Lux R is a transcriptional regulator that binds to the diffusing AHL back and in turn activates the transcription of its target genes. Okay. So, at low cell density AHL although is produced, it diffuses out into the extracellular space and lost, but at high cell density the concentration of AHL is so much, some of it diffuses back and binds to Lux R, which leads, as soon as they bind, this leads to a lot of transcriptional regulation that happens, like moving of the bacteria from sessile to fixed or biofilm forming form, production of virulent factors, activation of efflux pumps and so on actually. So, in gram negative bacteria this Lux I, Lux R play a very important role, and the AHL the signaling molecule gets diffused out and then again it comes back and binds to the Lux R. Okay. This is how the action of AHL happens in gram negative bacteria. Okay. What are the other biochemical factors which lead to bacterial resistance? Okay. You have the exopolysaccharides and glycocalyx. These are sugars and protein combination. Okay. So, they form a thick matrix on top of the bacteria. So, it delays or impedes the diffusion of antibiotic molecules into the deeper layer of the biofilm. Okay. So, they are more like a physical effect prevention. Then biofilm bacteria, they also activate many genes. So, their surfaces and other molecular targets also change. Okay. So, the antibiotics are not able to go and attach because their surfaces and other targets have changed, whereas the matrix, the thick matrix prevents the diffusion of the antibiotic, that is that we call it extrinsic resistance, whereas the changes on the surface and we can call it intrinsic resistance. Okay. Now, if you look at it from an engineering point of view, the diffusion of antibiotics through the, from engineering point of view, we can see the antibiotic or nutrient or oxygen diffuses through the biofilm from one end and reaches right up to the bottom. Okay. So, we can here use Fick's first law, you must have studied that long time back in your first year undergraduate. What is this Fick's first law? Flux is equal to, at steady state flux is equal to minus D, that is the diffusion coefficient, dC by dx. C is the concentration, x is the distance. So, dC by dx is the concentration gradient, J is your flux, D is the diffusion coefficient. Why this minus sign? Concentration of the antibiotic or nutrient could be is higher at the on the surface, whereas the concentration keeps decreasing. So, if you have a constant flux, we can assume this as a linearly decreasing concentration as we go deeper and deeper into the biofilm matrix. So, what does that mean? So, at the surface, if the concentration of the antibiotic is C naught, okay, as we go down, this concentration will be going down linearly and it will keep going down and down. So, at the surface the concentration could be MIC, whereas if you travel down it may be coming much lower, okay, and D sort of determines the slope of this linear line. Okay. So, if the D is very poor, that means if D is very small, so the fall in concentration as we go along inside could be very, very low. That means the concentration inside could be much, much lower when compared to the concentration at the surface of the biofilm. Okay. So, this D diffusion coefficient is based on many factors: how thick the biofilm is, what is the morphology of the biofilm, how much of exopolysaccharides that are produced. So, the diffusion coefficients can become very small. So, when it becomes very small, the concentration inside will be much lower than the concentration that is found on the surface. So, this is a very useful relationship to keep in mind, which tells you the mechanism or the reason why there is a huge fall in the concentration of the antibiotic as you travel inside this biofilm. Now, this is at steady state. You can also have an unsteady state diffusion, okay, where the diffusion flux and the concentration change with time also. So, we need to bring in the factor of time here. So, once we bring in factor of time here, it becomes a partial differential equation delta C by delta T. T is your time, is equal to d square C by dx square. So, we are considering only one dimension x. So, if you can have y, z, you can put in d square C plus d y square, d square C by d z square, and so on actually. So, it will be, it is a partial differential equation, that is why we put like this, whereas here we put like this. Okay. This is a ordinary differential equation, whereas this is a partial differential equation. So, the concentration is a function of time as well as the distance. Here it could be just x, but you can have x, y, z, all the three okay, coordinates also. So, here at time equal to 0, C is equal to C naught at x equal to 0 on the surface. At time equal to 0, concentration in the remaining, that means inside the biofilm will be 0. So, at time equal to 0, concentration inside the biofilm is 0. So, we the concentration of the antibiotic or any metabolite at the surface could be C naught. Okay. So, it is possible to solve this equation also. Let us not go too much into that and make you worried, but it can be solved, and you can have relationship that describes C as a function of time as well as x. Okay. Whereas, in the first law it is quite simple. When we have the constant flux J on the left hand side, then this dC by dx looks like a linearly decreasing concentration as we move along inside the biofilm. Okay. Now, we can have different situations of the flux and we can have different types of graphical observations. Let us look at it. Suppose we have fixed surface concentration, that is constant amount of antibiotic present on the surface of the biofilm, okay, that is fixed surface concentration. So, at the surface of the biofilm, the concentration of the antibiotics could be this much, but as you go inside the depth, concentration may be falling at a particular time T1. Then as the time keeps going up, it may slowly building up like this T2, T3 and so on actually like this. Okay. How fast it increases, that depends upon the diffusion coefficient and the depth of the biofilm and so on actually. So, if you have constant amount of antibiotic present on the surface, that means the antibiotic is not depleting constant amount. So, at the surface concentration will be so much. As you go inside the biofilm, the x axis is depth of the biofilm, the concentration will fall down like this, but as you increase the time, it will slowly build up like this. That means T1, T3 is greater than T2 greater than T1. Now, it is mathematically represented in this form: concentration as a function of x and t, Cs is the concentration at the surface, okay, constant surface actually, and this is how it is given. The relationship is given like this, okay, where d is the diffusion coefficient, t is the time, x is the depth. Now, you can have another situation, okay, where certain amount of antibiotic is placed on the surface of the biofilm at time equal to 0, that is fixed amount of dopant. That means the concentration at the surface also will decrease because as it keeps diffusing in. So, at very low time, the graph will look like this as a function of depth. So, initially at the surface you will have certain concentration, as you go inside the concentration will drop dramatically, but as time progresses the concentration inside may increase, but the concentration at the surface will fall down because we have fixed amount of antibiotic. Okay. So, as time progresses we are going to have like this. Okay. So, at the surface also it will go down, inside will slightly go up, and it is mathematically given by this particular relationship. Okay. So, we have two types of situations where we have fixed surface concentration, that is constant amount of antibiotic present, and we could have only some amount of antibiotic is placed on the surface. So, that gets also depleted as the antibiotic starts diffusing in. So, we have a graph as shown like this, whereas in the other case the concentration versus distance keeps going up and up with increasing time. So, as you can see from these explanation that the antibiotic amount inside the biofilm will be much lower than what is present in the surface. So, if the concentration at the surface is constant, then there could be a slow increase in the antibiotic inside the biofilm also, but if the concentration at the surface is also depleting, then we are not going to have a slow increase in antibiotic amount inside the surface. So, the bacteria inside the biofilm are always exposed to very low concentrations of antibiotic which could be below its MIC, hence bacteria starts acquiring antibiotic resistance. So, this is a physical phenomena which is based on the diffusions through a surface, and this can be explained both by using the Fick's first law as well as Fick's second law. Okay. Once more, as you can see, Fick's first law tells you the flux that is on the left hand side is equal to minus d diffusion coefficient dC by dx, and C is the concentration, x is the distance. Okay. So, it is a linearly decreasing concentration as a function of time if the left hand side is constant. Okay. So, at the surface concentration is high, so it keeps going down. But if you look at the Fick's second law, it brings in time as well into the picture. So, we have C the concentration function of time as well as the distance. So, we use partial derivatives here. Okay. dC by dt is equal to diffusion coefficients d square C by dx square. If we have the y axis also, then we will have d square C by dy square. If you have z, then it will be d square C by d z square. Okay. So, this is the Fick's second law which tells you that concentration can vary as a function of distance or as you travel inside the biofilm as well as a function of time as well. And from these laws we can develop certain pictorial visualization of how the antibiotic concentration could be distributed inside the distance as a function of distance as well as a function of time. So, in first situation we have a fixed surface concentration, that is constant amount of antibiotic flowing across the biofilm. So, at the surface concentration of the antibiotic could be very high, it falls down dramatically as you go inside the biofilm. This is at one particular time. As the time increases, this graph starts moving up and up and up and up, and ultimately the concentration inside could also be very high. That is when the antibiotic amount present on the surface is always constant concentration. Okay. And this is expressed by this particular mathematical relationship, whereas whereas in the other situation we have constant amount of antibiotics placed on the surface, and after that we do not replenish the antibiotic. So, it drops dramatically as you go down in distance, but because the antibiotic also starts diffusing, the concentration of antibiotic at the surface also keeps going down. Okay. So, at one time you will have a graph like this, as the time increases the graph may become like this and like this and like this and so on. Okay. So, the antibiotic is of no use, as you can see, beyond a small distance, and as you go travel inside you have very low concentrations which may lead to antibiotic resistance because the bacteria is exposed to sub MIC concentration of the antibiotic. And generally when a patient after implant surgery is given antibiotics, we can assume this type of scenario where initially the surface has large amount of antibiotics and afterwards the surface concentration of antibiotics also keeps going down. So, in the interstices or in the depths the concentration of antibiotic is below sub-MIC. Okay, so we will continue further on this biofilm and the effect of biofilm on implants. Okay. Thank you very much for your time.